All right, in chapter 10, part 3 for photosynthesis, we're going to talk about the light independent and light dependent reactions, and then we're going to kind of look at how the process of photosynthesis works from the light dependent reaction standpoint, and then kind of briefly get into the light independent portion. The much as we need to know as SL students, as well as HL, but the SLs need to know a little bit less, and then also talk about some of the similarities and differences between the mitochondria. So as far as photosynthesis occurs, the light-dependent reactions, these are the ones that are going to convert the solar energy into the chemical energy. Okay, and so the chlorophyll is going to help by absorbing the sunlight, and then we're going to kind of transfer that energy, and then that process is what's going to be responsible for generating the ATP. The electrons now in the hydrogen atoms that are kind of getting transferred from the water when the water is split to the NADP is what's going to give us some reducing power. In other words, what we're going to form from the NADP plus is we're going to get NADPH plus some H plus, and then the light reactions are going to generate this ATP that we need, and this is going to be some energy that's going to help us power these reactions. And it's important to remember that these things are going to occur on the thylakoid membranes, which are making up those stacks of grana. Okay, so as far as photosystems 1 and 2 go, it's important, one, to know that the photosystem 2 actually occurs first, and then it's photosystem 1. They're named in the order of their discovery, and what we get is we get some light energy. This light energy is being absorbed, and then it's getting transferred to this reaction center, and in the process of this we excite an electron. Okay, and since we're losing an electron down here, we replace it by splitting water, and here's the oxygen gas that we were talking about in an earlier part that we actually get. Okay, so the oxygen gas is given off as the byproduct, and now we have these electrons which are going to be able to flow down this electron transport chain, and when they do, that's what's going to help generate this ATP. Okay, so we're going to go in through all of this here. And then what happens in the light independent reactions is we have the Kelvin cycle that's going to bring in the CO2 from the air, and then it's going to form these organic molecules like glucose, C6H12O6. And that process requires energy, okay, in the form of ATP, which we've just talked about, and in the form of reducing power in the form of NADPH, okay? And these things do take place during the day, but they don't need light directly in order to function, and these occur in the stroma. Okay, so the light independent reactions occur in the stroma. The light dependent reactions occur in the thylakoids here. Okay, so we got that little problem ironed out here. So what we were saying is that the chloroplasts are the site of photosynthesis, and then we say that the light independent reactions, these occur in the stroma. The light dependent reactions are the ones that occur in the chloroplasts and the actual grana. Okay, so the light independent reactions, this is the Kelvin cycle. Okay, so what we've got here now is we're going to bring in some CO2, okay, and we're going to turn that into sugar, and with the help of the ATP in the NADPH from those light reactions. So if you look up here at this little kind of diagram right here, here's our light reactions. Here's where water gets split and the oxygen gas is given off as the waste product. These generate ATP and NADPH. Those get funneled into the Kelvin cycle, which you can see here, here, and here. And what they're going to do is help us take this CO2 and generate some sugar out of it. Okay, so these run through this first phase is called carbon fixation. This is where we bring the CO2 molecules in. We use some ATP to rearrange them, some NADPH to reduce them, and that's the reduction phase where we have this being reduced. It gives us glyceraldehyde 3-phosphate or G3P. That's what's going to get funneled into the sugar. We're going to use some ATP to rearrange the remaining glyceraldehyde 3-phosphate back into the RUBP that we're starting with. In the initial kind of as the CO2 is brought in, Rubisco is the most common enzyme probably on the planet that's going to catalyze the formation of this kind of first product here. And if you're an SL student, you don't need to know this. If you're HL, we'll go over this in a little bit more detail. So we've got the fixation phase, carbon fixation. This is where we bring the CO2 in. We've got the reduction phase. This is where we reduce it to give us CH2O, for instance. And then we're going to take, we've got 6 of these here. We're going to take 1 as an output. So if you look here, we've got 1, 2, 3 carbons. And this is our 3 CO2s that are being brought in. So we're left with 5 of these now. Okay, these things are going to get rearranged with the help of ATP into RUBP, which is going to allow this cycle to continue. So now that we've been through that a couple times, what I want you guys to see is we've got 3 carbons coming in right here. Okay, so we're going to form 3 of these short-lived intermediates. And then we've got 1, 2, 3, 4, 5, 6 carbons there. So 3 times 6 is 18 carbons. Now we're going to rearrange it a little bit. Now we've got 6 times 3, 1, 2, 3. We still have 18 carbons. Here we've got 6 times 3, so we've got 18 carbons. And then down here we've got 6 times 3, that's 18 carbons. We're going to pull out 3 carbons in the form of one G3P molecule. So now we've got 15. 5 times 1, 2, 1, 2, 3. So we've got 15 atoms. Here we've got 3 after the rearrangement. 3 times 1, 2, 3, 4, 5. So we've got 15. And then we're going to bring 3 more back in to give us the 18, and the cycle is going to repeat. So for every 3 CO2s we bring in, 1 CO2 is being given off. Okay? So excuse me, 1 sugar is being given off. So those 3 CO2s make the 1 component of the sugar that will get later recombined in a different portion of the plant to give us the C6H12O6 or the sucrose molecule, whatever it may be. Okay, so now as far as the photosystems are concerned, in the thylakoid membrane, this is where the chlorophyll molecules are going to be kind of organized into their respective photosystems, and this is what's going to allow photosynthesis to take place. So you've got these reaction centers that are surrounded by the chlorophyll molecules, carotenoids, different types of proteins. So we get some light energy kind of coming in, and once that light energy is absorbed, we excite an electron up to an electron acceptor, and then it's going to move down the electron transport chain. Okay, so now here's our reaction center, and you can see here's the thylakoid membrane that we were talking about. This is where the light comes in. Here's where, if it were photosystem 2, we would be splitting water. We excite the electron up to this electron acceptor, and then it's going to move down the electron transport chain. Okay, so there's 2 types of photosystems. We said they're named in the order of their discovery, photosystem 2 and photosystem 1. And when you're looking at them, they have a particular wavelength that they absorb light most optimally. P680 absorbs it, of course, at 680 nanometers. P700 is at 700 nanometers, and they're just absorbing some light. Okay, and that light is really what allows these photosystems to kind of work. They're basically the same thing. They just slightly have slightly different absorptive capabilities. And what they're doing is they're taking this light in, and then they're using, these photosystems are working together and using the sunlight energy to generate the ATP and the NADPH that the Kelvin cycle needs in order to function. Okay, so we've looked at this. This is where the electrons are getting excited. Okay, and then here's a diagram that we looked at earlier. So we've got this light coming in, okay, exciting an electron up to an electron carrier. It rolls down the electron transport chain and gives us this ATP. We have another kind of bit of light coming in, exciting an electron up to the electron carrier, and then it's basically going to move down an electron transport chain, and here it's going to generate the NADPH. So we've got ATP and NADPH from these light reactions. Okay, so these are the light dependent reactions, and it's going to funnel those into the Kelvin cycle. Okay, so the NADPH and the ATP will head to the Kelvin cycle. All right, so during the light reactions, there's 2 possible routes for these electrons to flow. You have cyclic and non-cyclic. Okay, so cyclic, basically what you have is the electron gets excited here, moves to an electron acceptor, moves down the electron transport chain. Once you get to the other kind of side of this, here it's coming down that electron transport chain. You have another photon of light exciting an electron up, and then it's going to move down this electron transport chain. So it basically goes to photosystem 2, down the electron transport chain, photosystem 1, down the electron transport chain. The first electron transport chain generates ATP. The second one generates the NADPH, which is the reducing power. Okay, so after they kind of transfer and flow down, this is what's going to give us the products that we need, the NADPH and the ATP for the Kelvin cycle to help make the sugar. And then with the non-cyclic flow now, that's where, you know, we're basically, this is the easiest way to kind of remember what's going on, is just it follows, if this is photosystem 2, this is photosystem 1, Basically, the electrons start here, move down a chain, start here, move down a chain. This one generates NADPH. This one generates ATP, and that's generally non-cyclic electron flow. Okay. And then if we have cyclic electron flow, basically what happens here is photosystem 1 works a little bit slower than photosystem 2. So in order to kind of keep up with the demands to make sure that we're efficiently using the ATP, what we can do is as these electrons start to get backlogged here at the top of photosystem 1, we can funnel them back into the electron transport chain that generates the ATP, so we're not wasting the energy. So this works a little bit slower than this one right here. And as a result of that, these tend to get backed up right around here. So we're going to funnel them back in. So you can see that this is kind of cyclic and that it continually repeats itself over and over again. Okay. Eventually, they're going to get out. But because we get kind of a backlog here and on the top of photosystem 1, we can funnel them back in. Back into the electron transport chain to generate the ATP. So if we look at this picture here, this is the thylakoid membrane. So you can see this is just kind of a tiny cutaway right in here. So we can see that if we blow it up a little bit, there's millions of these in a particular chloroplast. But what you got is you're absorbing the sunlight energy. Here's your photosystem 2. We excite an electron. We split water. That's the photolysis of water. The electrons transfer down the electron transport chain, and in the process, they pump hydrogen ions across the membrane. So this thylakoid space starts to get a very high hydrogen ion concentration. Okay, we have another photon of light, which is basically at the same time exciting in another electron, and this is moving down an electron transport chain, generating some NADPH plus H plus, so some reducing power. Now, between photosystem 2 and photosystem 1, we said that we're pumping these hydrogen ions across this membrane. They're ions, so they can't get across this nonpolar interior. They can only pass through one particular spot, and that's the ATP synthase molecule. As they pass through, as these hydrogen ions pass through, just like with cell respiration, they're turning a rotor, and that rotor is helping us take ADP and phosphate and giving us ATP. This ATP here is made, of course, into the stroma in the same way that the NADPH plus H plus is made into the stroma. Now these two things here are going to go to the Kelvin cycle to allow us to make sugar. Okay, so that's what we talked about earlier, is that ATP and the NADPH are going to get funneled into the Kelvin cycle to bring in the CO2, rearrange the atoms, reduce the atoms, and give us some sugar or some sugar precursor. Okay, so now, as far as some of the similarities and differences between mitochondria and chloroplasts, they both generate ATP through chemiosmosis, okay? And the electron transport chains are kind of in the membranes of these two particular organelles. And then what they're going to do is they're going to pass the electrons down and they're going to pump hydrogen ions across a membrane, either in the mitochondria, kind of into the inner membrane space, and then they flow back into the matrix, whereas in the chloroplasts, in the grana, they're going to go into this thylakoid space, and then they're going to be pumped out into the stroma, and that's where they're going to be generating the ATP. So in both cases, they're both going to take ADP and make ATP out of it. All right. And then the two mechanisms are very similar, but there are some differences between the two. Of course, with mitochondria, they're going to transfer chemical energy from the food to the ATP and the NADH, so the reducing power to allow us to make more ATP from that. And then the chloroplasts are going to transfer the energy from the sunlight to make the ATP and the NADPH. So one of them is going to use the ATP and the NADPH kind of as this chemical energy. The other one is going to generate it so that it can make chemical energy, okay, or do the chemical energy conversion, I guess would be the more accurate way to say this. One of the other differences here is we said that the hydrogen ions from the mitochondria get pumped into the intermembrane space, and then they flow back down into the matrix, which is where the ATP is made. In the chloroplasts, the process essentially reversed. What's going to happen is the hydrogen ions are going to get pumped into the matrix, or excuse me, into the thylakoid space, and then they're going to get pumped out into the stroma, which is where the ATP is going to get made and used by the Kelvin cycle. Okay, so This is just basically the words describing what I just said. Here, if we look at these pictures now, here's the mitochondria, the dark gray area. This is where the hydrogen ion concentration is going to be very high. So the chemical energy is used to pump the hydrogen ions into this gray area here. And then as it moves into the matrix, this is where the ATP is going to get made. Okay. And then over here, we can see this dark gray area in these thylakoid spaces. This is where the hydrogen ions are getting created, or excuse me, getting pumped to. And then as they flow back out into the stroma, again, this is where the ATP is going to get made. Okay, and then we can see really in both of these instances, these boxes, here's one, and here's one. This is the cutaway basically over here showing you what's going on. These hydrogen ions, as they're moving across the membrane, this is what's generating the ATP. Whether it's into the matrix or into the stroma, doesn't really matter. It's moving from high to low. All right. So here, if we look at these types of things, we can see that, again, kind of the similarity between the Kelvin cycle and these light reactions. These light-dependent reactions are generating these two substances, ATP and NADPH, that get funneled into the Kelvin cycle. Here we can see with the Kelvin cycle, the ATP and the NADPH get used to reduce this carbon dioxide into these sugar molecules. Okay, so I hope that helps you kind of understand the process of photosynthesis. Of course, if you have any questions, make sure you guys write those down. And then I'm going to go through a couple of other things that are HL only here for part 4. Okay, we'll see you soon.